Hi everyone, and welcome to our review on fibrous and globular proteins. So this is the next in our series of protein videos, and the final one in fact in our series of protein videos. What we're going to be looking at in this particular video are a couple of spec statements. So the structure and function of our globular proteins, including a conjugated protein, including hemoglobin as our example there, and a named enzyme plus insulin. Then we're going to have a look at the properties and functions of fibrous proteins with our three examples there: collagen, keratin, and elastin. So we have specific examples named in the specification for this bit, which means you need to know about these specific examples, folks. I haven't just picked them just for fun. These are listed in the spec, so we need to know them because they can ask us about these exact proteins. First thing then. Let's just go back into what we mentioned in the previous video about structure. So if you remember, we talked about those four levels of protein structure: primary, sequence of amino acids; secondary, alpha helix or beta pleated sheet; tertiary, three-dimensional structure; and quaternary, if it has more than one polypeptide chain, how they're arranged. What we find then is when we look at that tertiary and quaternary structure of our proteins, we can then divide them up into either fibrous proteins or globular proteins. So this is all based on tertiary and quaternary structures. We're going to start off with the fibrous proteins. Now, to begin, we've just got a few key points about fibrous proteins in general. Please learn these. The number of times that we get questions asking you to compare fibrous and globular proteins, it's a common thing. If you don't know these basic points about the proteins, you're really going to struggle with those questions, and they can be up to the 6 marker on this. So it really is worth making sure you know this well. So when we talk about fibrous proteins then, first thing, they're long chains of repeating patterns of our amino acids. They can form bonds with adjacent chains. They are insoluble, strong, and they've got this structural function. So if they're asking you for properties of our fibrous proteins, then you can jump straight in with insoluble. Insoluble is worth a mark on its own on pretty much any of these questions. So this is why this list is going to be really important. If they ask you about what the actual function of our fibrous proteins is, it's structural. So think about different structural uses of proteins. It's probably a fibrous protein there. Okay, and we're going to look at specific examples so that we've got those to call upon. Our first example then is collagen. So as far as the function of collagen goes, it provides mechanical strength. So don't just write it makes it strong. We're doing A-level biology here, folks. Let's use proper scientific terminology. Provides mechanical strength is what we need to be writing. In terms of where we find it, there's a whole array of places that we find collagen through the body. First place, in the walls of your arteries. Now, in those arteries, we find these layers of collagen, and the whole purpose of the collagen being present there is to prevent the artery bursting when it's under that high pressure caused by the heart's contraction. So if we didn't have collagen, the heart contracts, your arteries are then going to start bursting left, right and centre. Not going to be great for longevity. So, providing mechanical strength in our artery walls. Place number 2, tendons. Now, your tendons are the bits of the body that connect your muscles onto the bones. Now, if we didn't have something that was strong connecting those muscles to the bones, we wouldn't be able to move the bones very well, because when those muscles contracted, it would snap. And anyone who has experienced snap tendons will tell you that's not a good thing to have happen. So we've got that mechanical strength within those tendons because of the collagen, so that we can move our bones as those muscles contract. We also find collagen within the bones themselves, but this collagen's been reinforced with calcium phosphate to make it hard and bone-like. And then we find our collagen within connective tissues and cartilage as well. So what we can see, an array of different places of the body there that have our collagen, but the key thing that it's going to confer to all these different regions is mechanical strength. Second example, keratin. So what's the function of keratin? It provides mechanical protection, it waterproofs, and it forms this impermeable barrier to infection. So we've got 3 key properties there in terms of our keratin: mechanical protection, waterproofing, and an impermeable barriers to infections. So 3 key things to remember about keratin there. Why is it so good as this barrier? Well, it's because keratin actually has quite a large amount of cysteine. So it's what we call cysteine rich. Because we've got lots of cysteine amino acids present, that means we can form all those disulfide links or disulfide bridges, and therefore it's going to be strong. Because the more of those disulfide links or disulfide bridges we've got, the stronger the molecule will be overall. So what we tend to find then is that this keratin is found in any of the body parts that need to be hard and need to be strong. So think about your fingernails. They're made of keratin because we need that kind of hardness and the strength there to prevent the damage to, obviously, the tips of your fingers. In terms of other animals, then we find horns. Hopefully none of you have horns, but other animals do have horns, which are obviously going to be used in defense, etc. And therefore it's got to be strong, it's got to be hard. It wouldn't do a very good job of protecting you if it snapped at the littlest impact. Also see it in things like fur and scales, all coming back to the idea of mechanical protection. If we think waterproofing, fur there, think about those animals where water just runs off their fur. Keratin. Our third fibrous protein then is elastin. Now, the name kind of gives away what this one does, because if we think elastin, hopefully we're thinking that sounds like elastic, and it does. What this one does is providing the ability to stretch or change the shape of body parts. The reason it can do that, we've got a lot of crosslinks within its structure. So that means that the elastin itself is going to be strong and it's going to be stretchy or extensible. Best place that you'll find this is your skin. So if you actually just pull on your skin at the moment and let it go, you will find on the back of your hand, obviously, then you'll find that it just pings back to where it should be, assuming you've drunk enough water. If it takes a while to go down, have a drink. You're probably dehydrated. Now, what we find is your skin has this elastin present, and that means it can stretch around your bones and muscles. When you tense your muscle, that means that it's going to be able to stretch and change shape with that new shape muscle there. Always good. If we didn't have this, when you tensed your muscle, then it would be one of those kind of gruesome flesh ripping moments. Not pleasant. We don't want that. So we have our elastin allowing the skin to stretch. We also find it in our blood vessels. And again, go back to what we've said previously about arteries. There's that high pressure generated as the blood is contracted, as the heart contracts, forcing the blood out. We need our blood vessels to stretch and then recoil, so go back to their original shape and size. If they don't do that, if they just stretched and that was it, then our blood pressure would bottom out. It'd be terrible. So stretch and recoil in our blood vessels, down to the elastin. And one last region of the body where this is quite important is in the bladder. Hopefully we know the bladder is all to do with storing urine. It's one of those helpful bits of the body that stops you being rather smelly and gross by just leaving a trail of urine wherever you go. And the fact that your bladder can actually expand because of the elastin in that outside structure of it means it can expand to hold more urine. You don't literally have a fixed volume. As soon as you get to a couple of mils, that's it, job done, game over, everything else just leaks out, because that would be absolutely terrible. Instead, it can expand to a certain extent. Obviously, it's not an ever-ending expansion of your bladder, but it does expand to hold more urine, which then obviously lets you get to a toilet. At least I hope so. Next up, we're going to look at our globular proteins then. So 3 key points to remember about our globulars. Number 1, they are spherical. If you're ever asked about the shape of globular proteins, spherical is what we're throwing in. These are going to be soluble, so there's a key difference there between our fibrous and our globular. Globular are soluble, and the reason for that is they've got these hydrophilic R groups facing outwards towards that aqueous environment, and they've got this specific 3-dimensional structure, and that's going to be really important when we look at our examples. Our first example then of a globular protein is hemoglobin, an old friend from your GCSE course, or at least I hope so. I would hope we all know that the actual function of our hemoglobin is to transport oxygen around the body, and this is an example of a protein that has quaternary structure. So obviously it also has primary, secondary and tertiary, but it has quaternary as well as that. Now, what we find with hemoglobin is there are 4 polypeptide chains that make it up. 2 alpha globin and 2 beta globin chains. So within one hemoglobin molecule, there are 4 polypeptide chains, 2 alpha globin, 2 beta globin. Each of those 4 chains has their own tertiary structure, their own 3-dimensional structure. And what they're going to do is then fit together in a very specific way, and that gives us the quaternary structure with this very specific shape. What we find then is once we've got those 4 polypeptide chains all in our little structure there, we then find there's this additional little group, this thing called a prosthetic group. Now, this is actually a heme group, hence hemoglobin. This is where part of the name comes from. Heme, obviously referring to the heme group, the prosthetic group. Globin, referring to those globin chains it's made from. It's quite logical now we understand what it's made from. Now, that heme group contains an ion. This is going to be super important when it comes to actually transporting the oxygen. That's one of those things that confers this ability onto the protein itself. Now, because hemoglobin has this prosthetic group, this heme group attached to it, then we refer to it as a conjugated protein. And whenever we see this phrase conjugated protein, it's just referring to a protein that has a prosthetic group attached to it. So generally, prosthetic group, specifically for hemoglobin, it is a heme group, which contains this ion. When it comes to needing that oxygen for transport, we need some way to actually attach it to our hemoglobin. And this is where our heme group comes in, because the oxygen molecule is going to bind to the ion within each of those heme groups. So we have 4 heme groups within each hemoglobin molecule, which means we can bind 4 oxygen molecules. Note the word molecule, and hopefully we remember that oxygen as a molecule is O2. So if we have 4 heme groups, each one binding an oxygen molecule, each oxygen molecule is 2 atoms, that means that we can transport 8 atoms of oxygen per hemoglobin molecule. Now, when we actually see this binding of the oxygen, it brings about a colour change. So rather than having this very dark purpley red colour blood, which is what we can see in the little flask on the left there, okay, so that's our blood, that's the colour it looks without the oxygen bound to it. When the oxygen binds, it becomes this really bright red colour. Obviously, you won't see the purple red, because as soon as you injure yourself, and blood is obviously going to leave your vessels, then it's going to react with the oxygen in the air and become that bright red colour. So you won't see the purple red, but it is purple red, as we can see in that flask. Our second globular protein, as our specific example, is insulin. Again, something we should have heard about at GCSE and hopefully just in life in general. Insulin is a hormone. It's all to do with the blood glucose control. In terms of our description of the function for A level, it's going to bind to the glycoprotein receptors, and we look at glycoproteins in our membranes topic, on the outside of our muscle and fat cells, and when it binds those glycoprotein receptors, it causes them to actually increase the amount of glucose they take in. So that means we're reducing the amount of glucose circulating within our bloodstream, hence blood glucose control. What is insulin made from? 2 polypeptide chains in this case, so it has 2 chains, therefore that means it has quaternary structure, because remember, more than one polypeptide chain means quaternary structure here. We have our A chain, which has a little bit of an alpha helix at the beginning, and a B chain, which begins, conveniently, with a section of beta-pleated sheet. Isn't it nice when biologists actually name things in a logical way for once? So A chain, section of alpha helix, B chain, section of beta-pleated sheet. 2 polypeptide chains in our overall structure. Obviously, both of those chains have their own tertiary structure, their own 3-dimensional shape, and when we join them together, they're actually joining by disulfide links. We do find these hydrophilic R groups as part of the amino acid sequence. They will all be arranged on the outside of the molecule, therefore it is water soluble. Important when we're talking hormones, because how are they transported around the body? In the bloodstream. By being water soluble, it allows that transportation within the bloodstream. Our third and final globular protein that we will look at is our named example of an enzyme. So I'm looking at pepsin. You could have looked at other named examples of enzymes. As long as you know a named enzyme, you're all good. In terms of pepsin, this is going to be one of the enzymes we find in the stomach, and it is there to digest the proteins. So pepsin itself is a single polypeptide chain that's got 327 amino acids, which are then folded up into this tertiary structure held by hydrogen bonds and a couple of disulfide bridges or disulfide links. We do find that our pepsin has lots of acidic R groups. So it's got lots of amino acids that have these acidic R groups there. Because of that, it's really well suited to the acidic environment of the stomach. Hopefully we know the stomach is around about pH 2, so it wouldn't be great to have something that wasn't an acidic molecule really within the stomach, because it would just be denatured. Because we've got all these acidic R groups in its structure, it's perfect for that acidic environment. Reason for that? Because we don't have many of those basic R groups, the hydrogen ions in the stomach acid are not going to then bind and therefore influence its structure. Because they're acidic R groups, they don't have that ability. The hydrogen ions won't bind to an acidic group, only the basic ones, remember. So that means that the high concentration of hydrogen ions we find in the stomach will not have a negative effect on the structure there. So the enzyme keeps its 3D shape. Before we finish with our bits on proteins, we just need to understand a little bit about modelling. Now, when we talk modelling, we're talking about the ability to look at the shape of these protein molecules. We don't want to be in a situation where we're trying to look at them under microscopes and things like this all the time. Not feasible in all cases. So these days we can use things like computer models. Now, we can actually feed in information about the sort of sequence of amino acids, for example, and then we can use a computer to predict the shape of the protein from that primary structure. In terms of those secondary structures, it's all going to be based on basically probabilities of certain sequences giving a certain secondary structure. So they've analysed a whole bunch of different proteins, seen that that particular sequence of amino acids tends to create an alpha helix, for example, in 90% of the cases. So chances are if we see that little sequence, we're getting an alpha helix. So that's all been worked out from known molecular structures. And then we just apply it to our primary structure of the protein to make a prediction on what we could make. When it comes to trying to ascertain that tertiary structure, the three-dimensional shape of the protein, we've got a couple of approaches that we can take here. So the first one is what we call ab initio protein modelling. So here what we're doing is looking at the physical and electrical properties of the atoms in each amino acid within that sequence. Now, based on those, we're then going to make predictions on how they're all going to interact with each other, and therefore what shape will we get at the end. Now, this can result in more than one solution for the same sequence of amino acids. Option 2, comparative protein modelling. So this basically uses protein threading, where we scan the amino acid sequence against a database of already solved ones, and then it produces this set of models that would match that sequence. So what we're really doing there is just going through and saying, well, we've got these amino acids in this order. Previously, we've seen these amino acids in this order and it created that shape. So we think it's going to do the same thing. They're the two different approaches that we can take to obviously trying to predict the tertiary structure. Not perfect, as you can see, because obviously we've got multiple solutions by ab initio, and then the comparative protein modelling is just giving you a set of models. So again, not just a single solution here, there's potentially more than one outcome in each case, but it's a great starting point. As always, I do hope you found this video useful. Don't forget that on my website you can find a bunch of other resources that will hopefully help you in your A-level studies, and of course don't forget to subscribe so you can see when we upload another video.